Okay, so welcome to this next video in which we are discussing bradykinin-induced vasodilatation. Okay, right. So far we've seen that if we stimulate the endothelial cell with bradykinin, it will bind to the B2 receptors, which will lead to IP3 generation all over the cell, basically. IP3 will bind to and prime these IP3 receptors so that they are ready to open and release calcium from the endoplasmic reticulum if only they receive some calcium that will actually activate them to open. Okay, now something causes one of these IP3 receptors to get 4 calciums binding to it. So the first one gets 4 calciums binding to it. This could be from the cytoplasm potentially, or maybe there is a channel in the plasma membrane which allows calcium in from the extracellular fluid. Okay, but something causes one of them now to open. When it opens, it releases calcium in the vicinity of its location, and this calcium is going to spill over onto its neighbours and is going to activate them to open. They're going to release calcium, causing this little sort of calcium blip, as it's called, which is the calcium rise in the vicinity of an IP3 receptor that's just opened. And that calcium is going to spill over onto its neighbours, and basically you're going to get this propagating release of calcium known as a calcium wave. Now, it's a little bit more complicated than what I've described so far, because although calcium initially activates these IP3 receptors, when calcium concentration gets very, very high in the vicinity of an IP3 receptor, the calcium starts having a negative feedback effect. And this is not very well understood, but the calcium blocks the IP3 receptor from working once it gets to a high enough concentration in the vicinity of the IP3 receptor. So, initially, low concentrations of calcium activate the stimulatory calcium binding sites. The IP3 receptor releases calcium, causes this calcium blip, but as the calcium builds up in the vicinity of the IP3 receptor, it's going to negatively feed back on the IP3 receptor and cause it to close. So what will happen is this IP3 receptor will close here, and that will cause calcium to stop being released around there. So if we were to plot calcium concentration at position 1, position 2, and position 3 on a graph, then this might give us some intuition for what a calcium wave is, because it's more than just the propagating release of calcium. It's basically, you get a rise in calcium in this place, and then it goes down because of the negative feedback effect. And then you get a rise in calcium here, and it goes down again, and it's this propagating calcium spike, basically. And I should just explain why the calcium goes back down after the IP3 receptor has inactivated. It's because there is a pump in the membrane of the ER, which I'll draw here. So let's draw this in red here. So in red here, we have what's known as the sarco/endoplasmic reticulum calcium ATPase, which I'll write up here. So the sarco/endoplasmic reticulum calcium ATPase. Okay, and because that's a bit of a mouthful, people often just take its initials, which are quite nice, basically, ATPase. They take S-E-R-C-A, and they call it the Circa. So I'll write this down here, Circa pump. Now the Circa pump, if I draw it here again, okay, because it's a bit cramped up there. So here's the ER membrane, here's the Circa pump, which I'll draw in red here. Okay, so this is the Circa pump here. Then what the Circa pump does is it pumps 2 calcium ions into the lumen of the endoplasmic reticulum. So here come the 2 calcium ions, and it pumps 3 protons out every time it pumps 2 calciums in. So 3 protons come out. In addition, when it does that maneuver, it has to hydrolyze ATP, so it couples it to the hydrolysis of ATP to ADP, adenosine diphosphate, and inorganic phosphate. Right, okay, so these pumps are working continuously to return the calcium back in. So basically, when calcium is initially released from this IP3 receptor, it causes this calcium blip, and I'll write that down for you. So the tiny rise in calcium in the vicinity of an IP3 receptor, which results from that IP3 receptor opening, is what's known as a calcium blip, okay? So this calcium blip occurs, the calcium gets to such a high concentration that it starts inactivating the IP3 receptor. So the IP3 receptor closes, and then the Surka pumps return the calcium that's come out of the endoplasmic reticulum into the ER, basically. So that causes calcium to go down again. So let's look at this on a graph then. So if we plot calcium concentration versus time, so we'll plot calcium concentration up here, and we'll plot time down here. Then if we plot this for position 1, and I'll just remind you where position 1 is. Position 1 is over here. Okay, if we plot this for position 1, then what we're going to get is a graph that looks like this. Calcium is going to go up in the vicinity of that IP3 receptor. Then the IP3 receptor is going to be inactivated, so it's not going to go up any further, and then it's going to gradually go down as Surka returns it into the ER. So this is position 1. Now, let's plot the same thing for position 2. So position 2 is up here. Now, when the calcium goes up in the vicinity of the first IP3 receptor, we know that some of it is going to spill over and activate the IP3 receptor in the vicinity, well, in position 2. So what's going to happen is that the IP3 receptor in position 2 is going to be activated slightly after the IP3 receptor in position 1. So you're going to get a rise in calcium at position 2 later than in position 1. Okay, so I'll colour in position 1 in purple here. Okay, and I'll colour in position 2 in green. Now the same thing happens at position 2 as happened at position 1, basically. Once the calcium concentration around the IP3 receptor gets too high, then it's going to go from having a stimulatory effect to having an inhibitory effect. So the IP3 receptor becomes inactive, it stops releasing calcium, and the Surka starts returning calcium back into the endoplasmic reticulum, so it goes back down again, like so. Now finally, let's plot it for position 3 over here, and if we plot it for position 3, then position 3 is going to have its calcium spike even later than in position 2, because it's activated by the calcium overflowing from position 2, so we might draw it here. Its IP3 receptor opens after calcium has gone up at position 2, so this is position 2 here. And then again, the same thing happens, calcium gets too high, it starts having a negative feedback effect, and Surka pumps the calcium back into the endoplasmic reticulum. So this, finally, is position 3. Now that is one calcium wave, okay? It's this propagation of a calcium spike, effectively, across the endothelial cell. So, really what's happening is calcium is going up and then down here. It's causing calcium to go up and then down here. It triggers calcium to go up and then down here. So it's this propagating calcium spike, basically, that propagates along the endothelial cell. Okay, right. And that results from a global stimulation of the endothelial cell. Now, I want to stress that you don't just get one of these, because, you know, if your endothelial cell is being stimulated by bradykinin, the bradykinin isn't just suddenly going to go away, it's going to continue stimulating it, so you might have very prolonged IP3 concentrations in the endothelial cell. And what will happen is you'll get one calcium wave, that will propagate from one side to the other, and then you'll get another one, and then another one, and then another one, so you get repeated calcium waves. Okay, now calcium waves, the frequency with which calcium waves occur, i.e. how many you get in, let's say, a certain amount of time, it's determined by what IP3 concentration you have. Now this is not understood, basically. If you have a higher IP3 concentration, what we observe is that it causes more rapid calcium waves to go across the cell. Well, the actual calcium waves themselves aren't more rapid. A calcium wave is a calcium wave, understand that. But the gap in between consecutive calcium waves is going to be reduced, basically. I would compare it to neurons, and indeed it is always compared to neurons, where you have action potentials propagating along neurons. Now, a neuron can either fire an action potential or it cannot fire an action potential. It cannot change the speed at which it fires an action potential, and it cannot change the magnitude of an action potential. An action potential is an action potential. Similarly, a calcium wave is a calcium wave. You can't change the amplitude of these spikes. They are always the same, okay? You cannot change the speed at which they propagate along the cell, but you can change how frequently you have calcium waves. So you can change the time interval between consecutive calcium waves. And this is what I mean is dependent on IP3 concentration. So if you have a higher IP3 concentration, the time interval between consecutive calcium waves will be reduced, and lower IP3 concentrations mean a longer time interval between consecutive calcium waves, i.e. a lower frequency. So the intensity of these stimulus, i.e. how much bradykinin you tip onto this cell, that's going to determine how much IP3 you get. So the more bradykinin, the higher concentration of bradykinin, the higher the concentration of IP3. So the stimulus intensity is encoded by the frequency of calcium waves rather than by their amplitude. And that is exactly like in neurons, where the stimulus intensity, i.e. how hard you're pressing down potentially on a mechanoreceptor, doesn't determine how large the action potentials actually are. No, an action potential is an action potential. It's always the same size, but it determines how frequently that sensory neuron fires action potentials, and that's how you encode the stimulus intensity information. So it's very similar on this microscopic level to in neurons, basically. Okay, so, and the reason for that is because firstly this is the digital signal in this way. The stimulus intensity is encoded by the frequency of on-off signals, basically. So it's digital, it's more robust. And secondly, if it was encoded by analog signals, i.e. if the intensity of the bradykinin that you put onto this cell caused a greater spike in calcium, i.e. actually changed the amplitude of these spikes, then where would it end? If you could end up getting really toxic levels of calcium, basically, and that's the important thing to understand. We use this signal all the time in our cells. Calcium signaling is used everywhere, but calcium is an extremely toxic substance to cells. So you're really dancing with the devil, basically, and you have to be careful, and doing it through frequency rather than through amplitude is therefore understandable as to why that is a benefit. Okay, right. So there's the calcium signaling bit done. So we've got these calcium waves being produced in endothelial cells because of our bradykinin. Now, what is our calcium going to do now? Well, it's going to bind to a protein known as calmodulin, and this is then going to bind to the eNOS enzyme and activate that eNOS enzyme. So we'll talk a little bit about calmodulin, and then we'll talk about eNOS. Okay, so calmodulin then. The structure of the protein calmodulin in cartoons is often drawn like so. So you have these two lobes of calmodulin, known as the N lobe here, and the C lobe here, okay? And each of these lobes of calmodulin has two calcium binding sites. These are calcium binding sites. So the N lobe has two calcium binding sites, the C lobe has two calcium binding sites. Okay, and these binding sites are what are known as EFH domains, okay? So I will explain to you what an EFH domain is in a moment. So, an EFH domain then. So, an EFH domain is a polypeptide domain which is capable of binding calcium. Now, its structure looks like this generally. If this line represents the polypeptide, i.e. the polymer of amino acids joined together by peptide links, then it folds round in this loop like so. And basically, the calcium is going to bind in the center of this loop. Now, how does this polypeptide here allow the calcium to bind here? What does it do to get the calcium to bind here? So I'll draw the calcium in an orange color. Well, basically, the amino acids in this loop all have acidic side chains. So you pick acidic amino acid residues. Now, why would you want to do that? Well, let me draw you an example of an acidic amino acid residue, and I'll explain why from there. Okay, so let me draw you aspartic acid. So start with the general amino acid structure. So here's the carboxylic acid group down here, but of course this will be in a polypeptide, so this won't still be an acidic residue. It will be bound in an amide or a peptide link with this, the amino group of the next amino acid along. Okay, but if the R group is acidic, that will survive basically the process of being polymerized. So here is the acidic R group of aspartic acid. So this is an aspartic acid amino acid. Now, basically, aspartic acid is an acid because it is capable of donating this proton off this hydroxyl group of this carboxylic acid group. Now, when it donates that proton, what happens? So when this bond cleaves, what happens? Well, both of the electrons in that bond go to the oxygen. So the oxygen gets both electrons. Now, one of those electrons belonged to the oxygen, but the other originally belonged to the hydrogen. So that means it gets a negative charge. The proton, meanwhile, or the hydrogen nucleus, gets nothing. The proton goes off with a positive charge. So the point is that when the aspartic acid donates its proton away, the rest of the remaining molecule, which is known as the conjugate base of aspartic acid, or also it's known as aspartate. So that is the strict difference between aspartic acid and aspartate. Aspartic acid refers to the molecule when it's still got its proton. Aspartate refers to the molecule once it's lost its proton and has acquired a negative charge. It's what's known as the conjugate base of aspartic acid. This is the conjugate base. And it's called the conjugate base because it is now acting as a base. It will now act as a base. It can accept a proton rather than give up a proton. So it's certainly not an acid anymore. Instead, it's a base. It's known as the conjugate base of the acid. Okay, so the proton goes off, and the conjugate base of the acid is left with a negative charge. So if you put loads of acidic acid residues in this loop, so let's say these are the amino acids, if you put in a lot of amino acids which have acidic R chains, or R groups, sorry, then it means that they're going to gain negative charge, and that means that you're going to have a bunch of negatively charged R groups facing in to the middle of this loop, and those are what bind to this calcium ion because it's got a double positive charge. Basically, it's a divalent cation. So this is the calcium cation. Okay. Now, EF domains don't generally occur on their own in proteins. Instead, what they do is they generally occur in dimers. So I'll draw one of these here. So you generally have these dimers of EF domains where you have a linker region between them, and then you have another one sitting right next to it, and this is known as an EF dimer. Okay, so an EF dimer, and this is what happens in the N and C lobes of Calmodulin. Basically, you have these EF dimers, which means that you can bind effectively two calcium ions in one in each of these loops of the EF domain, but you combine them next to each other. And that's what you've got here. You've got EF dimers. You've got a single EF dimer on both of them, which means that overall you've got two EF domains and two calcium binding sites. Now, when Calmodulin does not have calcium bound to it, it is in this sort of a state here. The lobes are sort of bent over, hunched over towards one another. And this structure is known as apocalmodulin. Okay, so apocalmodulin. Or it's also often denoted apo and then CAM for short. So calmodulin is often denoted C, capital C, lowercase a, uppercase M for calmodulin. Okay, so this is apocalmodulin. Now, when calcium binds to the four calcium binding sites of apocalmodulin, what happens is it changes conformation. The two lobes of calmodulin move further apart, and the linker in between them goes from being a linear polypeptide to being an alpha helical structure here. Okay, so there's the alpha helical structure between the two lobes of calmodulin, which now have calcium bound to them in these calcium binding sites. So you've got calcium in these calcium binding sites here. And that means that the lobes spring apart in this way, and the linker between them gains this alpha helical structure. So this is an alpha helix, a secondary protein structure. Okay, and this structure now, where you have calcium bound to calmodulin, is known as a calcium-calmodulin complex. Calcium-calmodulin complex. Okay, and for short, it's often denoted Ca2 plus CAM. So, overall then, when calcium goes up in these calcium waves, what is going to happen is that we're going to produce these calcium-calmodulin complexes, and these calcium-calmodulin complexes are then going to go and activate our eNOS enzyme. But we'll continue this discussion in the next video.